Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last few classes we have introduced some of the recent NGS technologies, right. So, we have talked about Roche 454, we have talked about Illumina, and then in the last class we have talked about SMRT sequencing. In today's class we will discuss about two sequencing technologies in brief. So, one is called the ion torrent sequencing and the other is nanopore sequencing. So, that is the agenda for today's class. So, we will talk about ion torrent sequencing platform and we will talk about the principle of how this sequencing works, and then we will talk about the most recent sequencing method is called Oxford nanopore technology. Okay, so, these are the keywords that will come across. So, one is called semiconductor pH sensor, we will see this is relevant for ion torrent. We will talk about nanopores and picoampere current. So, let us start with the ion torrent sequencing method. So, ion torrent uses a semiconductor pH sensor technology. Okay. So, what happens is that there is a semiconductor sensor which can detect any change in H plus ion concentration through a change in current. Okay. So, this method also relies on synthesizing the complementary strand of a DNA molecule, right. So, again we have, imagine this is our genome fragment, right, and we have the primer binding site here and the bases are being added continuously. Okay. So, this requires polymerase and DNTPs, but here we need just normal DNTPs, no modified DNTP is required. So, what is the principle which actually is used by this ion torrent method? Okay. So, we will discuss this in a moment. So, here is the library preparation. So, it actually is very similar to what we have seen in 454 and Illumina. So, we have the genome here and we fragment it again into small pieces, and then we add the adapters A and B, right. This could be different, and again we amplify these fragments by some method and we do the sequencing. Okay. So, what happens in library preparation is that here we again follow a bead based amplification that we have seen in 454, right. So, if you remember from the 454 class, we talked about these beads where you have some oligos or probes that are already attached and the molecule DNA molecules come, they again they kind of prime, right, they kind of form this double strand and the complementary strand is synthesized, right. So, through emulsion PCR we actually generate multiple copies of the same DNA fragment on a bead, right. So, we follow the exactly same procedure here, right. We have again have these beads, we have emulsion PCR and we get multiple copies of the DNA molecule, right. And ideally if you remember from our discussion in 454 class, ideally each bead should contain, right, copies of a single molecule, right. But sometimes this might not happen and of course, those will give out confusing signals and which will then be filtered out later on during the data processing. Okay. So, we have the emulsion PCR step and we again get this amplification of the DNA fragments. Okay. Now, what happens is that the sequencing happens in these kind of picotiter plates that we have seen in case of 454. So, in these picotiter plates we have these, again we have these wells, right, and in these wells we have the beads. So, these beads will come here, they will sit on these wells and each well can accommodate only one bead, and we remember, right, each bead will have these kind of DNA molecules attached to them. Okay. And each well will have a pH sensor at the bottom of the well. Okay. So, as you can imagine, this method relies on detection of H plus ions, right. So, the change in the H plus ion concentration. Now, how does this H plus ion change then? Okay. So, this is something that is very important, right. So, when you have these DNA again, right, and here is the primer, you have the polymerase, it is adding this base. Okay. So, the moment one base is added, what you get is an H plus ion release. Okay. So, every base addition will lead to release of an H plus ion. Okay. So, if you look at the polymerization reaction, you will see this H plus ion will come in, and the detector then detects this H plus ion, right. So, the moment it detects an H plus ion, it will say okay, there is this addition of the some base, okay, and this way we can do the base call. Now, you might ask, right, how does it differentiate between different bases, right, different DNTPs, whether it is A, whether it is G, whether it is T or C, right, how does it detect that? If you remember, like 454, we supply DNTPs one by one, right. We do not put all DNTPs at once. We start with one DNTP, then we give the other, then the next one, and the last one, right. So, we have 4 going in cycles. Okay. So, this process is again same as 454, right. So, we have DATP, DGTP, DCTP and DTTP, okay, and this forms a cycle, right. So, one after another, and this gives us one cycle, okay, and you can again repeat this process as many times as you want, and you get a certain length of the reach in this data. So, as you can imagine, we can actually get very similar numbers as 454. Okay. So, as I just mentioned, we provide DNTPs in cycles, and in each cycle only one type of DNTP is provided, right. So, okay, so again now let us look at the sequencing reaction. So, maybe we can have a better understanding, and let us say we are following this kind of cycle, right. So, we are providing A, then T, then G and then C, okay, and we have this DNA strand, okay, and this is the primer and synthesis is happening, okay, and what we are doing is this is the cycle that is going on, right, and here is the signal, y axis is the signal, okay. So, this signal comes from the H plus ion, right, not light, here it is H plus ion signal, okay. So, what we do, so now you can imagine what we are doing, we are adding first DATP, okay, and we see is there any signal, is there any H plus ion detected by the pH sensor, okay. Let us imagine there is, okay, and we say okay, there is this addition of A. Then we let us say give E, and so here is we have added, then we give T, right, and we see okay, there is no signal, okay, which means T has not been added, right. So, the first base is A, then we add G and we see okay, there is signal, okay. So, this means the sequence is AG now, okay. Then we add C, right, and we see okay, there is signal. So, then we see, and we repeat this process, right. So, now you will have A, okay, again, and you see if there is signal, then T, G, C and so on, right. We can continue as many times as we want. Of course, there is a limit because of the polymerase that we are using. Now, one of the things you probably realize that again like 454 you have a washing step, right. So, you give one DNTP, right, A, you see wait for signal and then wash away everything and again give the next DNTP, right. So, it requires a washing step like 454 because we are giving one DNTP at a time. So, you need this washing step, okay. Now, so you can look at this video which will give you the overview of the whole process. This is an animation and you will get a better idea of the whole process, okay. So, what are the advantages of this method? So, this method gives us very similar read length as 454. So, it is about 400 to 700 base pair. It has reasonably high accuracy. So, as you probably remember 454 was also reasonably accurate and it is about 99% accuracy that you can get. So, which is good. It is also quite economic, right, compared to the competition, right, because you do not need any optical detection in it, which could be a bit expensive and it require kind of a higher capital cost for that machine, whereas here you need less capital cost, right. So, the machine is not so expensive because you do not have any optical detection in it, you have a semiconductor based pH sensor which is more economic, and also we do not need any modified DNTP, right. So, this also reduces the operating cost of the system. What are the drawbacks, right? So, one of the first drawback is that you have lower throughput compared to Illumina platform, One of the things you will notice, the maximum we can get is 100 to 130 million reads, okay, and this is much less compared to Illumina data that we have seen, right. We have seen some statistics for Illumina where you can get billions of reads, and here it is only about 100 to 130 million at most. And it also varies from chip sequencing chip that we are used that you will use here, right. So, again it depends on you can see some of the chip names and you can see the throughput that will get from these chips. Again, depending on your application, you will pick the right one, right, whichever will be most suitable for it. So, one of the things you probably can notice that this is probably not really suitable for large genome sequencing projects, right. So, if you want to do let us say a huge project, genome sequencing project for large genomes, you probably would not choose this method. But on the other hand, you can think of, right, if you are working with small number of samples and small genomes, this is probably the ideal and most cost effective method that you can use. There is another drawback, right. So, and this is actually shared with 454. Okay. So, one of the things you probably now remember, right, in 454 we have this signal that is proportional to the number of bases being added, right. So, if I can again draw this signal versus cycle and we are following this ATGC cycle, right, and imagine let us say we have first A we are detecting, okay. So, we have detected A, now we see T and what we see is the signal is much higher, okay. So, which means we have probably added 2 T's here, okay. So, this signal the sensor that detects this signal, right, the signal should be proportional to the number of bases being added. So, you can imagine that if you have 2 bases you have 2 H plus ions, right, that are being released. If you have 3 bases added you have 3 H plus ions. So, the signal that will be detected by the sensor should be proportional to the number of bases being added, okay. So, then we have G and maybe we miss G and then again for C maybe we see higher signal and we can say okay we have 2 C's added here, okay. Now, the problem is beyond 8 to 10 base pairs this signal intensity is not proportional to the number of bases, okay. As we have seen in case of 454 this tends to saturate, okay, and which means beyond these 8-10 base pairs in the homopolymeric stretch regions will not be able to accurately determine the length of the stretch, right, and will also have more indels or insertions and deletions that will be detected, okay. So, this is a problem where we are not doing synthesis of this complementary strand one base at a time, right. So, we see that this is not there in Illumina, this is also not there in PacBio SMRT sequencing, but here we have this problem again because the principle is very similar, right, like 454, but I mean the signal detection is different where we, in 454 we detect light, here we are detecting the change in H plus ion, right, but we are using unmodified DNTPs, right, and we are allowing addition of multiple bases at time. Okay. So, these are some of the sequencer names that are available, again they vary based on their throughput, but the principle is same, right. So, you have ion gene studio S5 or ion torrent gene access. Okay. So, let us now move on to the final sequencing technology that we will discuss. It is called Oxford Nanopore technology, or in short ONT. You will see this term ONT in many places. So, this is a method that actually relies on direct DNA sequencing. Okay. All the methods that we have discussed so far, starting from 454, Illumina, then to SMRT and then ion torrent, they all require synthesis of a complementary strand of the DNA molecule. So, this method does not do that. Okay. This method directly sequences DNA molecules, and this is what makes it incredibly powerful, right. So, it means it simplifies everything, right, the library preparation, etc. You do not have to do any amplification, you do not have to generate any clusters, etc., right. You do not have to have any polymerase in there to synthesize the complementary strand, okay. And this means this method is likely to be more economic than all other methods that we have discussed. It also means this method can sequence really long reads, it can generate really long reads because you are not limited by the processivity of the polymerase that you are using for the synthesis process. Okay. So, let us now move into this method and see how this actually achieves all this, right. So, how does it do the direct DNA sequencing. Okay. So, the library preparation, as I said, is very simple, right. So, we have this genome which is fragmented into smaller regions. Again, although this method can sequence really long reads, it cannot sequence the full genome at once. Of course, we will like to do that, but that is still not possible. So, you need to fragment it to smaller fragments, right. You need to get smaller regions, and but they can be much larger than all the methods that we have discussed so far, right. So, Illumina or Ion Torrent or 454, where you need smaller fragments, here you can have quite large fragments which can be then sequenced. And then we have this hairpin adapter ligation. So, it is a very simple step, there is no amplification required, you simply add this adapter and then you can start the sequencing. Okay. So, what happens here? So, is something like this. So, you have a nanopore like structure. So, you can imagine a flow cell, right. So, the sequencing happens inside flow cell, is kind of, you need something like this, right, either SMRT cell, flow cell we have seen, or picotiter plates, right. So, here you have a flow cell where the sequencing happens, and on this flow cell you have millions of these very small pores, okay, and these are called nanopores. Okay. So, they are actually a small structure that are embedded on kind of a matrix, okay, and these nanopores are made of proteins, right. So, these are very small dimensions, okay. So, only one strand of DNA molecule will pass through this. So, single stranded DNA molecules, they pass through the nanopores, okay, and when they are passing through these nanopores, they are being sequenced. Okay. So, we will see this in a moment how this happens. So, as I have just mentioned, you have the matrix, okay, and you have these nanopore like, so these are the nanopore structures where you have these very small holes or pores, right, and you have a protein, right, that will separate out this double stranded molecule. It will just direct one strand through this, right, the other strand will be going outside. Okay. So, only one strand will be passing through this, right. This is a protein that will do this job. Okay. Now, how do you actually detect these spaces, right? As you said, this is a direct DNA sequencing. So, how do you actually detect this? Okay. So, it turns out there is a current that is applied across this nanopore. Okay. So, it is a picoampere current, right. So, very small amount of current that is applied through this nanopore. Now, when these DNA bases are passing through the nanopore, they actually lead to some change in the current, okay, and this change again is dependent on the sequence of DNA that is passing through the nanopore. Okay. So, you can imagine this situation, right. So, if we are drawing this time versus current, so y axis is current, right, this is in picoampere. So, very small current. So, let us say we have this kind of constant level of current that is passing through the nanopore, and then at this point we start passing the DNA molecule through the nanopore. So, what will happen is that because of these specific sequences, right, they will modulate or change the current, okay, and we will start seeing this kind of peaks, valleys, some sort of change. Okay. Again, this change is dependent on the sequence. Okay. So, by looking at the change in current here, right, what kind of change we are seeing, we can then identify, okay, what are the bases that are passing through the nanopore. Okay, and there is a specific characteristic of this specific set of sequences that are that will induce certain changes in the current. Okay. Now, as you can probably imagine, this is a quite a difficult process, right. So, identifying which bases are passing through the nanopore from the current signal. So, one of the questions you might have, okay, is this detected by single base? The answer is actually no. This is actually detected by a pattern or combination of bases that are passing through, right. So, a combination of base will have different patterns, and you can find these unique patterns from these combinations. Okay. So, I hope this is clear how the general principle how nanopore will detect the DNA bases. Okay. Here are the two links where you can actually see some animation that will make the whole process clear to you, and you will get a better understanding. So, please have a look. So, what are the advantages? So, one advantage is that this is a long read sequencing, it is a really long read sequencing, because you are not limited by the processability of the polymerase, you are detecting the DNA sequence directly based on the change in current, and you can imagine you can continue this process as long as you want, right. There is no polymerase required that actually is doing the synthesis and it will stop at certain point. This is not the case. So, you can keep on passing DNA molecule through the nanopore, right. So, that ideal that is what we want, and that is what that is where we are going, right, where you can probably sequence the full genome at one go. So, we get long read sequencing. So, this helps in genome assembly, and really long ones, long reads will also help in identifying the long structural variants, the big ones that are present in the genome. So, we can achieve read length of 10 to 100 kb, right. You can imagine, right, you can see this compared to the other methods, this is actually quite long. Okay. In addition, as you have seen, like SMRT, we have real time observation of the current, right, change in current that is happening, right, that is the signal, and we have real time observation what is happening to the current with respect to time, and this data is stored in the data file that comes from Nanopore. So, when we talk about these data formats, you will see we have this data in there, and this data is stored, and that can be quite useful for specific applications. In addition, this is also a single molecule sequencing, right. So, we are getting the signal from single molecule, right. Like SMRT, we are sequencing one molecule, and each nanopore will get a unique DNA molecule. So, this is something again really advantageous, because we can now identify what are the changes in the DNA molecule just beyond the basis. Okay. What if there are some other modification or some specific characteristics, those can be identified because we are now observing these single molecules in real time. Okay. So, you can see these two classes of methods now, right. So, we have like 454, we have Illumina, we have ion torrent, where we need an amplification step and we get the average signal out, right. So, the detectors are detecting the average signal from multiple clones of a DNA molecule, right. So, that is why the amplification step is there. In case of SMRT and in case of nanopore, what you see now is that there is no amplification step, we have detection of signal from single molecule, and we have real time signals. Okay. So, this is really valuable for different applications. And one of the biggest advantage of nanopore sequencing is that this is really economic. As you can see, right, you do not require any optical detection device, you do not need any polymerase, you do not need any DNTP, which means the cost comes down a lot, right. So, compared to other methods where you need this synthesis process, right, even if you compare now with SMRT, right, which is a single molecule real time sequencing, but that requires synthesis of the complementary strand. Compared to all the methods, this is the most economic and most cost effective compared to all sequencing technologies, right. So, this is why this method will get popular and will probably be the sequencing of the future. Now, one of the questions you might ask, right. So, when we discuss that Illumina is probably the dominant platform today, right, most sequencing that happens in Illumina. So, the question is, why do not we shift to the nanopore sequencing? Why are not we doing sequencing in the nanopore? Because it is really great, right. We have long reads, we have like low cost, etc. Everything is there in favor of this method. So, why are not we switching? So, here comes a drawback, right. So, the error rate is actually very high. Okay. So, if you compare to Illumina or other methods, right, 454 or ion torrent, this error rate is quite high, right. So, it is about 20% error rate. Okay. Now, the good thing is you can actually improve upon this error rate, and by method called ONT 2D. Okay. So, we have so ONT stands for Oxford Nanopore Technology in short, right. So, ONT 1D, right, where we actually have the DNA molecule, okay, and you can imagine, right. So, in as we have mentioned, again we have this hairpin adapter. So, we can sequence only one side, right, of this, because these are complementary strands. So, double stranded DNA, right. So, we can sequence pass only one side of the DNA molecule, right, this protein that is present at the entry of the nanopore, right, it will pass only one side and we can detect, okay, this sequence. Okay. So, that will be ONT 1D. Now, what we can do, because we have this hairpin adapter, we can also sequence the other side, right. So, you can see, you can kind of move these and move back and forth, right. We can go through this, then we can again sequence the other side. Okay. Now, again this will help in improving the accuracy, because we are getting sequence from the same DNA fragment, right. These are complementary strands, but the same part of the same fragment. So, we will get more sequence data, and this again helps us in calling the consensus sequence. Okay. Right. So, this hairpin adapter allows us to sequence of both strands, and this is called the WNT2D, right. But what we have seen, what researchers have seen, even WNT2D has high error rate, which is about 13%. Okay. So, what can we do about this, right. So, because this 13% error rate is very high if we are interested in especially identifying mutations or genotyping, etc. So, what has been happening in recent years, that there has been improvements in accuracy, and one of this comes from more number of detectors being added in the nanopore. So, earlier in the nanopore method, there was only one detector per nanopore, right. So, you can imagine this situation, you have the flow cell, you have these nanopores, and each nanopore had only one detector. Now, there are two detectors, they are slightly separated from each other, right, which are detecting this change in current. So, they are being affected by a larger combination of bases, right. So, as you increase the uniqueness of the bases, you actually can get better correlation with the signal change or the current change that is happening. So, more number of detectors actually lead to higher accuracy. So, these are some of the sequences that are available. So, again these are based on different throughput levels. The principle of sequencing is same. So, the first one is called SMIJAN. It is a very small sequencer. You can actually connect it to your smartphone, and you can do the sequencing and get the data on your smartphone. And then you have this sorted according to the throughput, right. So, you have MinION, you have GridION and Promethean. So, what about the potential of the nanopore technology? So, one thing you can see, right, this is a direct DNA sequencing, and perhaps that this can have many more applications just beyond DNA sequencing. So, one of the first thing we see, we can actually also do direct RNA sequencing. So, we will talk about this in transcriptome analysis, right. So, when you do the transcriptome sequencing, we want to measure expression levels of genes. We first have to convert the RNA that we isolate into cDNA before we can apply any of this earlier sequencing technology. But with nanopore, you can actually directly sequence RNA, right. Here there is no amplification, etc., no primer binding, etc., required. So, you can simply again kind of set up the process for sequencing RNA directly. So, one of the things that will change is that this RNA bases, the signal, the current signal change will be different, right, and that will have to again take care of when we are detecting the bases, right. When you are doing the base calls through the change in current, we need to have program that will identify those RNA bases. Okay, but that is a solvable problem, right, and we can do that. So, we have seen recent improvements in accuracy, and it has actually reached about 99%, and hopefully it will go even further now. It is a real time sequencing, right, and we have seen this is direct detection of nucleotide modifications we can do. Again we will talk about this in much more detail because of the real time nature of the signal, and we are also detecting signal from single molecules. And we have I have told you, right, we can do RNA sequencing, but it also turns out that we can also sequence proteins. Now, there have been some reports, some work from some research groups that are showing that we can actually sequence proteins directly using the nanopore technology. Now, one of the things you can now visualize, right, in your mind, because the principle is so simple, right, there is no synthesis required, the principle is based on change in current due to the chemical nature of the molecules or the atoms that are passing through, right. So, you can ideally should be able to do this protein sequencing as well because of the principle, right. So, proteins again different amino acids will generate different patterns, right, or the combination of amino acids will generate different patterns for the current that is flowing through the nanopore. Okay, so, recently this has been applied and has been shown it can identify even single change in amino acid in a stretch of protein. Okay, so, here is the reference where you can see the application of nanopore sequencing for protein sequencing, and can also identify this difference in single amino acid. So, please go ahead and have a look at the paper, and you can you will now understand, right, the potential of this Oxford Nanopore technology. So, here are the references that we have used for this class, and to summarize, we have talked about two sequencing methods in this class. So, the first one was ion torrent sequencing. The ion torrent utilizes a semiconductor pH sensor, and it detects changes in H plus ion concentration when the sequencing reaction is going on, right. So, when you synthesize the complementary strand of a DNA molecule, there is release of H plus ion every time a base is added, okay. And this change in H plus ion concentration is detected by the sensor, which then calls the base, okay. And this happens in cycle, which we have discussed, right. We do 1 DNTP, we add 1 DNTP at a time, and we observe for signals. And we have seen we get reads that are comparable in size to Sanger sequencing, but we have again the problem that we have seen earlier. There is higher error rate in the homopolymeric stretch region. So, we get more indels in those regions. We then talked about the nanopore sequencing technology, and this is a technology that is entirely different from the other technologies that we have discussed. So, this method directly sequence DNA molecules, and the detection happens through the change in current, right. So, again a stretch of or a combination of base will change the current in a unique way compared to other combinations, right. So, this detection actually helps in direct identification, and this is real time detection, right, at the single molecule level, and it is very much cost effective because we do not have any polymerase or DNTP involved. But what we have seen, we have higher error rate, and this is being addressed now by addition of more detectors, also doing something like 22D, right, or consistent sequencing, and this will actually help in a better accuracy. And we have also discussed, right, this is a method that has a lot of potential for the future because it is cost effective, it is doing the sequencing by direct detection. So, it can be applied for RNA sequencing as well as protein sequencing, and we have I have given you some references for further reading. Thank you.